Hello students. In this presentation, we will be discussing about the mitosis cell division. If we talk about the definition of mitosis cell division, we can say that this is the part of cell division which results in two identical daughter cells which are formed from a single parent cell. Talking about the various stages of mitosis cell division, it is divided basically into 6 stages which are as follows. The first stage is known as the interphase or the resting phase. Second phase is prophase, in which the cell is going to prepare itself for carrying out the division process. Then comes the metaphase, followed by anaphase. The second last stage is telophase, and the last stage is cytokinesis, in which the division of cytoplasm will occur. Coming to the first stage of mitosis cell division, it is the interphase, in which the cell will be preparing itself for carrying out the process of division. This stage is common in both plant and animal cells, and in this stage, the DNA will be replicated, or in short, we can say that the DNA will be copied. It will make new organelles, that is the synthetic phase, and the cell will grow in size. The growth will occur. If we see the diagram of the interphase, we can see that there is a nucleus present, followed by the nucleolus, which is present inside the nucleus. The chromatin material is present, which is bound by a nuclear envelope. At one part of the cell, two centrioles will be present, and the cell membrane is the plasma membrane. After this comes the prophase, in which the cell will prepare for nuclear division. Both in the animal and the plant cells, the chromosomes will start appearing. The centrioles will separate and they will form the spindles. The nuclear envelope will gradually disappear, and this is considered to be the longest phase in the mitosis cell division. Here in the diagram, we can see that in the nucleus, the chromosomes, they contain two sister chromatids. They are bound by means of the centromere. There is the aster formation, and a pair of centrioles are present which will move apart to the either of the poles. After this comes the metaphase, in which the cell will prepare the chromosomes for carrying out the division. In the animal and the plant cells, the chromosomes they line up at the middle of the cell. That is why it is called as the metaphase. The spindle fibers attach to the chromosomes at the centromere, and this is considered to be the shortest phase of mitosis cell division. In the diagram, we can see there are formation of spindle fibers. These spindle fibers will be connecting the chromosomes by the means of centromere. This is the metaphasic plate, and the chromosomes will align themselves on the equator. After this comes the anaphase, and the chromosomes will divide in this phase. The spindle fibers, they will be pulling apart the chromosomes, and half of each chromosome, called as chromatids, are going to move to the poles for cells. You can see in the diagram, the cell has become oval in shape. The daughter chromosomes, they are gradually moving towards the poles, and they have parted off into two arcs. After this comes the telophase, in which the cytoplasm will divide. This phase will be considered separately in the animal cell and in the plant cell. In the animal cell, the DNA will be uncoiling, and again it will be appearing as chromatin again. There will be formation of two nuclei, and the cell membrane will pinch off into form two new daughter cells. Talking about the plant cell, the DNA will be uncoiling, and again it will be appearing as chromatin material. Two nuclei formation will be there, and new cell wall forms between two nuclei to form the two new daughter cells. We can see in this diagram clearly there is a development of cleavage furrow. The nuclear membrane has started appearing, In each daughter cell, the chromosomes are there and there is gradual disappearance of the spindle fiber. The cytokinesis, which is considered to be the division of the cytoplasm, and this will create two new cells. Again, it will begin in telophase, and in animals, if we want to uh, identify this stage, we will be looking for a cleavage furrow. Thank you.